Dear students, today we will discuss about the disease amoebiasis. The disease amoebiasis or amoebic dysentery is caused to man by a protozoan parasite called as entamoeba histolytica. Entamoeba histolytica, or it is also called as dysentery amoeba. It was discovered by Lambe in 1859. The disease is caused to human beings all over the world, but it is widespread in tropical and subtropical countries. Entamoeba histolytica. The parasitic protozoan Entamoeba histolytica belongs to phylum Protozoa, class Rhizopoda, order Lobosa, genus Entamoeba, and species Histolytica. It is a monogenic parasite, that is, it completes its life cycle in one host, that is man. In the previous chapter, we have already discussed about malarial parasite. Malaria parasite was a digenetic parasite, but Entamoeba histolytica, it is a monogenic parasite because it completes its life cycle in one host, that is man. It is amoeba-like in appearance. A full-grown trophozoite measures about 20 to 50 micron in diameter under the microscope. It is seen as a colorless and a transparent mass of protoplasm surrounded by a thin elastic plasma lemma. The cytoplasm is distinguished into outer ectoplasm and inner endoplasm. The endoplasm contains nucleus and food vacuole also. Life cycle of Entamoeba histolytica. The life cycle of Entamoeba occurs in three stages, that is trophozoite, pre-cystic stage and cystic stage. Trophozoite. The trophozoite is the most active stage of the parasite. It is the most active stage of the parasite. The trophozoites secrete a tissue dissolving enzyme which helps in dissolving the mucous lining of the intestine and in penetration into the intestinal wall. The trophozoite secretes a tissue dissolving enzyme which helps to penetrate into the intestinal wall. It feeds the RBCs and intestinal tissues of the host cell. Inside the intestinal wall, the trophozoites divides by binary fission and form flask-shaped ulcers. Then pre-cystic stage. This is the pre-cystic stage. Some of the trophozoites, after being discharged from the ulcers into the lumen of the intestine, enter into the pre-cystic stage. When the trophozoite, after discharge from the ulcer, they enter into the pre-cystic stage. At this stage, the trophozoites are round and small. They are called as minutiform. Their cytoplasm contains one or two chromatoid bodies, glycogen, vacuoles, but no RBC. The cytoplasm of the pre-cystic stage of the minutiform contains no RBC. Then the last one is cystic stage. During unfavorable condition, the minutiform becomes covered by a thin transparent refractile cyst wall and change to cystic form. In this encysted condition, the nucleus of the individual divides into two daughter nuclei and then to four, that is tetranucleated stage. The two daughter nuclei stage is called binucleated stage. Then to four daughter nuclei called tetranucleated stage. These cysts are highly resistant to desiccation, temperature, chemicals, and are the infective stage of the parasite. These cysts are passed out along with feces of the host and appear as small shining ball. The cysts from the outside reach the gut of the new host through contaminated food and drinks. The cystic stage of the parasite of the protozoan, when discharged from the fecal matter of the human being, then it enters into the new host through contaminated food and drinks. This is the diagrammatic sketch of the life cycle of Entamoeba histolytica. Life cycle of Entamoeba histolytica. Life cycle of Entamoeba histolytica. This is the structure of trophozoid. This is the nucleus. This is the ectoplasm and inner endoplasm. This is the outermost wall, plasmalemma. Then the trophozoid divides into two daughter individuals by binary fission into young trophozoid. Then it forms the cyst wall. Then binucleated stage, 2 nuclei. Then 4 nucleus, tetranucleate cyst wall formation. Cyst, tetranucleate stage. Then encysted also occurs. Then the metacyst trophozoid released from the cyst wall. Then they enter into the trophozoid. This is the life cycle of the life cycle of the Entamoeba histolytica. Then mode of infection. A healthy human gets infection when the cysts are ingested by him through contaminated food, drinks, uncooked vegetables, fruits, etc. Either directly or through the agency of housefly and food handlers indirectly. This is the mode of infection of the parasitic protozoa. Symptoms. In the intestine of the man, the trophozoid secretes a tissue dissolving enzyme which help it to penetrate into the intestinal wall and form class-XII ulcers. From these ulcers, contents are discharged into the lumen of the intestine so that blood and mucus are passed out along with stool. In severe cases, passing out of the mucus and blood along with the stool takes place. In severe condition, at an interval of every few minutes. It is the severe condition of the parasite, of the patient. The patient suffers from abdominal pains, headache, nausea, etc. Nausea means vomiting stage. Diagnosis. Amoebiasis is diagnosed by examining the stool of the patient, showing the symptoms of the diseases in positive cases. The cysts can be detected in the stool under the microscope. Then, treatment. Although it is not easy to control amoebiasis, still several drugs are in use for many years. The drugs are emetine, terramycin, bioform, lidocaine, enteroquinol, mexaform, etc. There is no immunization method against this disease. There is no immunization method. Tinidafil plus and Tinilux MPS is much effective in eradicating the disease. Then prevention or control measures. Following methods should be taken for prevention of amoebiasis. Protect. Number one is protect the food and the drinking water from contamination. The uncooked vegetables should be soaked in 5% potassium permanganate solution for about half an hour before use. Hands should be washed properly, washed with soap before taking food. Drinking water should be purified. Human feces should be chemically treated. Educating people about the simple hygiene of the disease. So this is the about disease amoebiasis. Thank you.